So maybe plankton will be relevant to you, even if you think more about physics. Okay. First, thanks to many people who have contributed thoughts and content, etc. First, if you can all either go to this website, don't think you'll need the code, but or scan this on your phone so you can do laptop or phone, and you'll have a little survey, and it's going to ask what comes to mind when you hear phytoplankton community composition. Does it work?
Yep.
Great. I'll give you 45 more seconds. I'm getting typed and I'm gonna switch to the other screen. Happy? Okay. Stand by while I get the results. No, I unplugged it. It's good. Oh, I was like, results are inconclusive. Cool. Okay, ready? Everyone happy? Will it work? So we have pigments coming in at the top, taxonomy, diversity, algae, bad, biodiversity, carbon, chlorophyll, coccolithophore, cute, cyanobacteria, DDAS, not sure, diatoms. I'll read them. Dynamics, ecology, functional traits, HABs, hot mess, inversion, mesoscale features, microphytoplankton, mix, ocean critters, pace, PFT, size of species, trophic succession, zooplankton community. Fabulous. Love it. We'll go back to the PowerPoint. And I think perhaps one thing that jumped out is that there are many aspects to phytoplankton community composition, perhaps even invoking the hot mess comment. This seems problematic.
I think it was the connection.
Yeah, but it's not as. I'm trying to remember why that. It's sort of
Unplug and replug.
Yeah.
Well, we'll see. Okay, well, diving in here, some past results from different classes. So kind of fun to see the overlap and the differences. This is the class from 2 years ago. And another class held in the European class from the year before that, where climate change jumps out. It's interesting, a little bit of a different take from that class. But lots of different responses kind of spanning both the biology and the ecosystem features, but also some of the approaches and strategies that we use. All right. This is a great PACE image from a year ago, the Gulf of Maine. Who has an idea for what determines these different colors this year? This is a true color image, but the gains have been tweaked to brighten things, but true color. Near true, I forget what they call them, Jeremy. Near true color image or something. Just yell things out or whatever. Why do we see all these different shades?
Phytoplankton.
Definitely phytoplankton.
Sediment.
Sediment, yes, for sure.
Sedum.
Sedum. Currents. Eddies.
Outputting.
Upwelling.
Atmosphere.
Nice.
Cool. Okay. Good answers. I also want to point out that you have various shades of green and teal, and it's even more impressive on your computer. So when you have the slides shared, you can see on your machine, but this, what looks like a dark area, is actually like a dark green. So all these various shades of green and various shades of brightness are the result of different types of plankton and the cetone and the sediments. So it's a muddled problem, but I think this is a nice clear example of how it's not just greener water is more chlorophyll and bluer water is less. And I'll emphasize that further by showing the same image, but the chlorophyll content. Now that dark green area really pops out as high chlorophyll. Right, so we have both brightness and color affecting our ocean color, true color image, and then chlorophyll. And perhaps that's because the chlorophyll that we see is the result of different types of plankton that are contributing to it. I will say, let's just say almost definitely. And is there something we can do to know which types are there? I mean, spoiler, there's not going to be a next image showing which ones are here, but that's what I've been banging my head against the wall, and many in our community, and maybe some of you want to tackle this challenge too, of understanding what's behind this chlorophyll symbol. Also recognizing that some of these coastal places, Bay of Fundy, there's cetone and sediments that are certainly impacting our estimates. So zooming now down to the in situ level. Again, it's a little clearer on your screen, but I think you can see here the difference in the color of these two water of the water in these two images. There's lots of skyline here, so it starts to get blue, but looking closer to the ship here, you can tell it's greener blue, and these are pictures taken just a few days apart on a transect off the coast of South Africa with extremely strong upwelling and a strong gradient of chlorophyll. Just a little bit of a spoiler, but here are IFCB data, and we have many more plankton in our greener water. Fewer, but still some happy diatoms and ciliates in the bluer water. And then the filtered water has this amazing red color, and that's due to some of these very happy dinoflagellates, probably.
Is it filtered water?
Yeah, concentrated.
Oh,
Yeah, sorry, not filtered. Yeah, concentrated. I'm not sure how much volume, but yeah. And it's also interesting to note that like these plankton that may have this color, like maybe you saw it in the culture room here, the different colors when they're in culture and very, very concentrated, that then turns our ocean into yet a new color when we're seeing the effects of absorption of water. So another example, these waters both look pretty blue. Maybe you can notice that this one's a little bit different. And these are waters with, with the caveat, the IFCB isn't looking at the phytoplankton, so we're just thinking about the medium and large plankton here, but kind of similar in terms of the number of particles. But on this side, it's a diatom blue, and now we have this amazing yellow brown concentrated water, and that's the diatom color. So what motivates our interest in trying to parse out which types of plankton are in the water from the color of the water, from ocean color remote sensing? The top left figure shows variable carbon flux as a function or as it relates to particle size distribution. And then here's a schematic showing how variable phytoplankton size types can be in terms of size with a analogous scale with images of objects that we know. Maybe you've seen this before. It's a popular one to show. Try to wrap our heads around the extreme diversity in phytoplankton in the ecosystem and what that means for carbon flux sinking, food webs, higher trophic levels, may or may not want to eat some types of plankton and others, and obviously that propagates up to maybe fish or species of interest or direct interest for human activities. And of course, on a global scale, we want to know how is our Earth system changing.
How is carbon flux calculated? With just a ratio applied to it, or like?
These are casts, and I'm not from, so these are all stations from the Tara Global Expedition. Maybe Gil knows.
So the exports is calculated using the UEP data. So it's just taking pictures of particles, and then there is a simple equation. Model what is actually what is coming from the top and actually top profile. So these are all profiles from Tara.
Maybe BB too? No, I don't know. Maybe just UVP?
No, the equation is just based on the top of the. Yes, exactly. So based on bi-volume, assuming some, based on 2D images, assuming bi-volume, and then assuming.
But like from floats to typically be calculated from BB as an estimate for phytoplankton particulate carbon. So looking at vertical profiles. And yeah.
Just these profiles, they were matched with sediment trap to have a relation between the estimates from the UVP
and the rate, I guess. And this is really Meg's domain. I don't see her, but she's a sediment trap. She has her and with Wayne. Well, and maybe Niels and Patrick. Instrument to measure the rate of particulate flux.
There's many different ways that we can try and measure it, and everyone thinks that their way is better. There's lots of discrepancies between methods and how well they work.
Gotcha. But it's based in essence on biovolume, when we say carbon flux here.
This one is, but others will be based off of, so the same filtering setup we do for chlorophyll, you do it a little bit more cleanly, but you just filter for POC onto the exact same filter. You can bust that filter and you run it through a mass spec and you can get the exact amount of carbon. And then you can relate that to backscatter and then get a different measure.
And that would be like a carbon concentration per volume filtered. You know, I mean, you would know the concentration because you know how much you filtered. Like a biomass. Yeah.
Very
Mass.
organic carbon we're talking about.
Usually. People also
Yes.
measure that.
Yes.
That's
And
okay.
and Kath Mitchell, who'll be back tomorrow, is a PIC expert.
Yeah, so the whole over one side. We've got the bird settings, mountain sheet, one that's clear and that's mountains.
Yeah.
So maybe get to this.
All right. We could go all day, I think, discussing uncertainties. So, and this is a paper from Heidi Dearson and colleagues, kind of an overview paper where they're highlighting some of the areas that we can study using hyperspectral remote sensing. And I just put stars where phytoplankton community composition plays a role in some of these parameters. So it's relevant in kind of a wide range of applications. But how are we defining PCC? Phytoplankton size classes, also PSC. Also, you might see that sometimes is the acronym for photosynthetic carotenoids, which are a group of pigments. So lots of P's and S's and C's. Phytoplankton size classes are typically defined as picoplankton, nanoplankton, and microplankton. And in the literature, there are some consistencies there, but also what size cutoff should we be using for those groups? Phytoplankton groups, maybe a catch-all term. Phytoplankton functional types, maybe a little bit less popular than it used to be, but it's still used. Are we talking about biogeochemical function when we use the word functional? And buyer beware, the meanings of all of these terms may change based on the user in the paper, etc. So bottom line, we want to define the phytoplankton in the water by some metric that differs or moves beyond the total biomass, which we typically approximate via estimates of chlorophyll A concentration, but not always. But in some way, we're trying to move beyond total chlorophyll A. How do we measure PCC in situ, which is necessary to develop our algorithms and understand what's actually there so that we could maybe try to approximate it with optics and remote sensing? Microscopy, pigments, flow cytometry, automated imagery, so like imaging and flow cytometry like the IFCB, merging size spectra from multiple flow cytometers to try to build a PSD that spans all of our phytoplankton size classes, optical signatures, obviously. Now molecular methods becoming much more common in recent years to use to characterize the phytoplankton community. And what about units? Are we talking about absolute units, concentrations, biovolumes, biomass, chlorophyll A, the contribution of a certain group to chlorophyll A? That's a relatively common way to describe the plankton community. What does that then mean for carbon flux or food web dynamics? Relative units, also fairly common. So the proportion, as I just mentioned, a group contributing to chlorophyll A or to total biovolume. Sometimes you see the dominant group, maybe probability of occurrence, at what threshold. All of these are valid and useful, and just to be aware that you'll see all of these in the literature. And Sasha and colleagues did a study with data from multiple cruises where they had measurements of phytoplankton community composition done several different ways. So pigments, RNA, molecular analysis, and flow cytometry, both IFCB and FCM, which refers to flow cytometry, like the conventional flow cytometry, typically quantifying pico and some smaller nanoplankton. So also just to notice that even when we try to match up how we're defining phytoplankton using different methods, there are discrepancies. Some results from that study. This first plot is comparing the larger eukaryotes, so pigments on the top versus 18S and IFCB. You'll notice maybe two things. One is that there do seem to be some consistent trends. The darkest color, so on the bottom, darker red, which is actually brown on my computer. But that's diatoms, and there's a pattern that matches in terms of their contribution to the total. But also you'll see that we're measuring different types with different methods. Similarly, looking at the picoplankton, flow cytometry, and the smaller stuff and the larger stuff with the molecular analysis, and pigments for all groups again. And the colors are a little harder to see on the screen, but Prochlorococcus has some consistent patterns throughout, but challenges. Okay, so now we are trying to define our phytoplankton community in situ. It's possible, but it's good to be aware, especially if you're harvesting a data set for somewhere for some subsequent analysis. How is it assessed, and what are the uncertainties and caveats to whatever method was used? Because really what we're trying to do is scale up our phytoplankton community composition so that we can have our remote sensing view of a basin or a region or the world, and understand our vast ocean ecosystem. But it's a trade-off, of course, between the direct measurements we make with our in-situ discrete samples and the spatial and temporal resolution of a satellite image. And kind of at an intermediary between our discrete in-situ samples and our remote sensing is continuous measurements made on the ship. And we did this on Tuesday, so it'll be fun to look at that data. But this can be a nice link where we get an insight into the spatial variability of some of our measurements that we don't get from the discrete samples, but we do have better constrained and higher temporal spatial resolution, than our satellite and remote sensing images. So shipboard continuous measurements are a powerful tool for algorithm development and other studies. This is an image from a Patrick Gray et al. Paper showing schematically how all the different ways that we try to assess the ocean have trade-offs in their resolution, but also their coverage. And I just like this image, so maybe you all do too, just to kind of see everything all at once in terms of what we're trying to measure and how we're doing it, and how those two challenges interlace. And kind of another way to view that same information, this is a slide adapted from Mary Jane Perry, showing that we have our remote measurements, satellite, aircraft, drone, yes. And those are the measurements and we're trying to invert them to get at our parameters of interest. So we're motivated by understanding our Earth system, harmful algal blooms, maybe fisheries, measurements that are relative to human life, understanding our ocean, but we need to link them to what we're actually measuring. And right at the middle, joining these two disparate ends is optical proxies and algorithms. So I'm a product of a manuals lab, so I'm heavy on the methods and algorithm development. That's really something he has a lot of expertise in. That's what I think about a lot in my work, so that'll be a theme throughout, but of course it's also important to be considering all the different pieces of this flow. Traditionally, algorithms to detect different aspects of the phytoplankton community from remote sensing fall into two categories. One is abundance-based. So we're going to estimate chlorophyll A, as Jeremy explained this morning, from our reflectance. And then from there, we're going to relate the chlorophyll A concentration to the PCC empirically. Or we can look at the spectra, maybe derive IOPs, maybe not, but try to relate somehow our spectra, both remote sensing, reflectance, or IOPs to the phytoplankton community. Abundance-based phytoplankton size classes. Lots of people have worked on this. This is an example of figures from Bob Bruin paper. He's actually several papers on this topic. But here is an earlier one showing kind of just looking at chlorophyll on the x-axis of these plots, and then the size fraction on the y-axis. So nano, pico, and nano, and microplankton. And you'll notice that picoplankton, the fraction of picoplankton contributing to the total phytoplankton community decreases when chlorophyll goes up. Inversely, we have more big stuff when there's more chlorophyll. So on a global scale, this is our first order relationship. More chlorophyll, more big stuff. And that's shown nicely here. You can see the microplankton fraction is high where we have lots of productivity, high latitudes, upwelling regions. And inversely of high picoplankton proportion in the gyres. We used some in situ data also to test this relationship. The previous plot also used pigment in situ data, but here we're using in situ data from cytometry, so merged IFCB and flow cytometry to see how these relationships play out on a smaller data set, but perhaps measuring the plankton sizes more directly, certainly measuring the plankton more directly with cytometry rather than pigment proxies. And we see some of the same relationships, but definitely noisier. Abundance-based, so now this is a related approach, but rather than looking at size classes, we're going to compare chlorophyll to phytoplankton community composition as defined by pigments. Put that in bold here, something to keep in mind. So we have all these phytoplankton groups, and they've been defined using HPLC pigments, which includes caveats, but we'll come back to that. And so you can see there are these kind of global scale relationships between the total amount of chlorophyll and these the contribution to chlorophyll of these different phytoplankton groups. So there's also, that was abundance based. Now we have spectral based. So this is, in this case, looking at phytoplankton absorption spectra. On the left are in situ data. This is work by Artichiati and Inika Rico. They have several papers developing this method and applying it. And the basic idea is that your phytoplankton, normalized phytoplankton absorption spectra shape changes when you have different size stuff in the water. So over here in the upper right, you have higher SF, which is their size fraction metric when you have smaller plankton, and that's due to a steeper slope because you have less plankton packaging when you have more small cells. So this is and then, yeah, from CWIFs. So they applied the method with in situ, developed the method with in situ data, and then applied it to remote sensing to try to get a sense of spectrally derived size of the phytoplankton community from space. Now, spectrally derived taxonomic groups. So this is one of maybe the earliest, the 2005 paper looking at phytoplankton groups. Calculated, estimated using spectra. So these are just multispectral data. The groups are defined using pigments in situ. And then the map is showing the dominance of a given phytoplankton group from remote sensing. So this is kind of a early days trying to map phytoplankton groups from space. Since then, zooming ahead to this decade, pre-land who's technically still at NASA? No. Pre still at NASA?
No.
Okay, well, she wasn't, this was developed when she was
When she was.
working there, but she's still involved in this kind of work. So this is an algorithm that Bree and colleagues developed to estimate the concentrations of different picoplankton groups using a data set from the Atlantic Ocean, which is why it's been applied just in the Atlantic. And this is a principal component analysis and using in situ flow cytometry data. And this is now termed the Moana algorithm, and it's available as a PACE product. It was one of the early demo pictures from PACE, demo images from PACE, but I don't know. I haven't looked at the data since it's been being produced, so I'm curious too. But I don't know. Yeah, it's available as of very recently. So if anyone wants to play around, it's just available in the Atlantic. But if anyone wants to play around with PACE-derived phytoplankton and picoplankton groups, that would be cool to see. And there's a review paper by Ivana et al. In Remote Sensing of Environment from 2024. And it has a table if you're interested in really digging into the literature and you want to see what has been done. This is hopefully near comprehensive ways that we're trying to extract PCC from remote sensing data. And also just to point out that the algorithm validation data varies widely, right? So kind of circling back to the numerous ways we can even define phytoplankton in the first place, and then what might be available for a given algorithm development effort. A slightly earlier paper from Mao et al. They show this table, and I just want to point out here, you see our algorithm development inputs and our satellite outputs. Two things. One is that HPLC pigments is a frequent, appears frequently, and that our inputs don't match our outputs. So this is a fundamental algorithm limitation. We're trying to develop an algorithm using in situ data that actually isn't the same as what we're then estimating, which when we phrase it that way, sounds somewhat concerning. But we do know that there are relationships between these things and our satellite outputs, but it's good to keep in mind when you're using a product from satellite that there are limitations based on how it was developed. As I just pointed out, phytoplankton pigments show up a lot. That's fully logical based on our amazing global data set of HPLC pigments that the Royal Hour, for data sets spanning the globe, and much of that pigment data is publicly available. So pigments have been used in numerous studies to define the phytoplankton community. But there are some limitations, which is that pigments do not necessarily equal our cellular carbon biomass, and this relationship varies widely. The taxonomic resolution is limited because you have pigments that are not diagnostic to certain groups. And typically phycobilly proteins are excluded from HPLC data. And pigments come from discrete samples. So thinking about some of the gaps in our efforts to estimate PCC, we have an information mismatch, which you saw between what we're trying to estimate and what we have to work with. Perhaps a lack of robust uncertainties in some areas and uniform way to trace uncertainties through algorithm development. Missing capabilities, I would say this one has been and continues to be addressed, which is very exciting. And regional capability. So an area that could expand infinitely in different situations for, when there are motivations to develop regional algorithms. This certainly has lots of room for growth. And about 20 years ago, I mean, around then, obviously it was starting a bit before then too, but the idea of using hyperspectral data to get more information than our traditional multispectral that started in the late 70s was kind of coming on the scene. So this is an oceanography magazine paper showing at the very fundamental level, look, we're missing this information if you don't have multispectral. So can we add hyperspectral to the mix when we're thinking of gap filling and getting more information? I guess you've heard about PACE at this point. That's a simulation, but I guess there are some real videos now, like the launch one, which is incredible to see. But yes, of course, now we have this near continuous measurement of ocean color throughout the visible. And that provides a lot more information.
Maybe this is obvious. But like, I understand the hyperspectral will be giving us more channels of information, but if our methods haven't changed, how, I guess I don't understand how it's gonna help us.
Yeah, cool question. The question is, if we have more channels, but the methods haven't changed, like if we're still using a band ratio algorithm, how does it help us? We do need new methods. So we'll kind of come to that in the talk, but methods that take advantage of the whole hyperspectral suite of information that can only be done hyperspectrally, and then obviously would be good to compare them to heritage methods. But there's opportunity there. We've done some the community has done work on that, and there's room to grow. So for those of you who are intrigued by this, you can tackle your own new hyperspectral methods, and that would be great. I don't know if you want to chime in, Jeremy, in terms of value added beyond approaches that require, full suite of hyperspectral inherently, or PACE also is unique because it has a polarimeter. So unfortunately, I'm not going to talk about polarimetry today. Patrick can speak to polarimetry and community composition. I should have included this, but I think they've already seen the slide with the DOLP and the IFCB group. So.
And I'm new to this, I have never looked at this So I'm just curious, I guess, so
Yeah.
what you guys think.
Yeah, I mean, for me, at a fundamental level, kind of going back to this, this is that we can potentially tease out information from spectral features that we just didn't measure. So if we have 100 channels instead of 5, we'll be picking up features in the RS that are due to absorption, differential absorption by different pigments. But we'll come to that. It's gonna, yeah. Well, I'm biased by my phytoplankton group absorption feature aspect of why hyperspectral data are. We'll talk about a couple of little things, but yeah. Other people might have other ideas. So this is a review paper that Ivana led, and this is just kind of a fun image. There was a survey that went out to the community, and you've all met Ivana, so it tracks, but these are some of the options on the survey. Anything as long as it's not chlorophyll only, every species out there. And this is an answer to the question, what do you want from PACE in regards to community composition? So the paper is serious and has lots of good info, but also this image is fun and speaks to the diversity of needs of the community and the end users, and how there's not a one-size-fits-all algorithm that we're developing for PCC, community developing. Yeah. But taxonomic groups jumping out as a leader. Okay, and also this review paper that I mentioned earlier, but there's a nice categorization of different ways that we can extract information. So kind of answering your question, what information can we get out of hyperspectral data that maybe is unique to the added content from so many channels. So I'm not going to go through all these different types, but just discuss a few of the ways that the community and also in my own work, we've tried to extract information from hyperspectral data. Okay, so apparent visible wavelength, or AVW, is a method. There's a paper from Vandermeulen et al. In 2020, and then also a follow-on paper, Dearson et al. In Frontiers 2022 maybe, using the AVW as a QC metric for hyperspectral data. But this is a method that calculates the weighted harmonic mean of every spectrum. Basically, you can think about it as a way to diagnose the color of the water based on all of the content in the spectrum. So it's considering the reflectance at every wavelength, and then it's calculating the mean, but weighted harmonic mean, so that higher wavelengths don't have more impact based on the fact that they're higher wavelength. And you can see that the AVW value changes, as you move from blue to green water. Sometimes I've seen a version of this where the color bar is more intended to reflect the true RGB color, but you get the idea here. And here's a difference between a delta between AVW calculated using multispectral MODIS aqua data, which are the blue lines, and hyperspectral HYCO data, which was a hyperspectral ocean color imager that flew for some period of time on the ISS. No longer there, but there are some years of data from HYCO, that's imagery over coastal regions. And you can see that the apparent visible wavelength, there's a difference when we use hyperspectral versus multispectral to calculate it. Because, well, there's also inherent differences in the two sounds. The point is that when we have the hyperspectral spectral resolution, we end up estimating a different AVW. So maybe we're honing in a little more on the color of the water when we have more wavelengths. Way to think about that. Here's the AVW that I calculated for the Puget Sound. So this is in the west coast of the US, where I live and do some work. And there is a sensor. It's going to come up later, so I'm going into it now. But there's a sensor called EMIT with some hyperspectral opportunistic data that I played with a little bit in coastal regions. So this is my attempt to match AVW values to the real color. 550 is green, etc. So you can see over the Puget Sound is like a coastal fjord kind of water body. That there's greener water in these central basins and bluer on the side, and then right close to the coast, you have some orange and yellow water. So sort of experimental, but also what pops out as interesting is that when we look at the relative amount of photoprotective carotenoid pigments, which is something that plankton synthesize to deal with highlight, like a sunscreen, you can see that there's looks like we have higher relative PPC in bluer water. So this was just something to play with, but kind of a fun little demo application of AVW, and you can use it. Other ways that we might use the full hyperspectral suite of information. This is work by Nima Palivon. I'm saying right? Palivon. And also Ryan O'Shea, who's now picked up a lot of this effort. This is the suite of Aquaverse products that's in the works to be implemented as the PACE hyperspectral product. So this takes the hyperspectral RRS and calculates probability density functions for all of the inverted parameters. So kind of a leveling up of what Jeremy explained this morning with our inversion algorithm. Now we're going to calculate the probability per wavelength of a given value. And they've got some machine learning in here too. But they train this with a lot of coastal data. And so they have a trained model now that they're deploying in coastal and also like some lakes and inland water. And looking at a phytoplankton absorption at one wavelength, you can see that the mixture density network comes up with a tighter relationship. Some of the heritage from them. So in this demo, in complex coastal water, that's kind of a major value added of this approach. Everybody doing okay? You just stretch, drink. Am I talking too fast, too slow? Cool. We're doing all right. Big fun time. Okay, so really digging further into why do we even expect there to be differences? Yeah. Yeah, now we're on, yeah,
It's not the Phytoplankton.
It's not, you won't see it, it's
looking at satellite images, if we don't need the lights out. Something is relevant. Yeah.
I don't know if now or later in the talk is a good time to ask, but what does the and I don't know the name of the community, but like the arboreal community, what do they do for like do they use hyperspectral imagery for identification of tree species?
Yes, for sure. And there's even someone on the PACE there's even some PACE work on that, even though it's designed as an ocean observing mission. There's some land-based hyperspectral algorithm development to separate different types of trees. So over land, they have a lot more signal in the red. So they use the red edge information, which is yeah. Say it again, son.
No, I might have been, but it's where in vegetation it's giving a yeah.
So what's the red they reflecting
In the infrared. Yeah. So you have a strong reflecting signal in the NIR. Around 700. Okay, so just at the edge of the red visible. And as with the multispectral for ocean, traditionally there's been multispectral going out into the red and the NIR. So you might have your red edge is like connecting two dots, but now it's a hyperspectral signal. So I forget exactly what they're doing, but fitting or something to try to see subtle differences in the reflectance out in those red wavelengths, and then using like, in situ data from the land to do similar work, but for types of vegetation.
And
Yeah.
phenology is a lot more useful because trees just aren't green. They change over time, and how they change is very specific to the species. So like being able to see those colors with fine detail helps with that.
And I know this sounds daft. Over the course of a season, this is me not knowing, the phytoplankton also change their pigment compositions.
Yeah, potentially based on nutrient limitation or light limitation, not so much the suite of pigments that a given species will contain, but the relative proportion.
Yeah.
You might synthesize more for protective, or they might struggle to synthesize their accessory pigments if they're nutrient limited. Sort of the edge of my plankton physiology species knowledge, but yes, I would say as a broad brush answer, but it's not so straightforward as trees, obviously, that aren't moving, so then you're trying to tease apart water masses, populations, also the plankton are living for a day or two versus the tree, so you have all these other dimensions of. Yeah, but yeah, potentially now that we're starting to look at pigments, not just total chlorophyll, maybe there's some seasonality and relative pigments for a given group. Just to complicate the goal. Okay, so pigments absorb at different wavelengths. I guess maybe this has come up already in this course, but it's this information that is at the crux of what we're trying to, well, for me personally, a lot of what we're trying to do is tease apart this information from our hyperspectral data, both in situ absorption data and in situ remote sensing, reflectance, and then remote sensing from satellite data, and tie it all together. But does it become ill-posed? We have a lot of pigments. We have uncertainty in our measurements. So this is a paper led by Kale, and there are other related papers, but Kale will be here next week with his advisor. I think it's gonna be. Here you can see on the left that you have 7 pigments, and then on the right you have a lot more pigments, and this was some theoretical work to show that if you want to extract a lot of pigments from one spectrum, then you need very, very highly constrained uncertainty measurements. And your error, your uncertainty, will determine your degrees of freedom and how many different pigments you can separate. These are just some takeaways from that paper that apply to that in situ absorption data set, but also apply more broadly when we're thinking about our uncertainty in our measurements and what that means for our degrees of freedom. So if you have a lot of uncertainty and error in your measurements, that's the difference between having a few degrees of freedom rather than dozens. In some cases, there's nothing we can do. Our uncertainty is our uncertainty, but usually if we've done our science right, the uncertainty is the uncertainty. It's not going away, but we need to know what it is, because that will determine our degrees of freedom when we're thinking about inversions. In some cases, we can reduce the results of the impacts of our error if it's just noise that can be averaged out. But if we have a bias or high environmental uncertainty, there's nothing to do about that. Most of the variability in our ocean color is due to chlorophyll A. But then we start to have other dimensions of variability that are sort of stacked on top of that. And so when we think about going beyond chlorophyll, we're trying to tease out how things are changing in the ocean and differently varying relative to chlorophyll, even though to first order, many things in the ocean vary either directly or inversely with chlorophyll. Maybe polarization can help us tease out some of these variable relationships in total chlorophyll. UV information, potentially useful to separate CDOM and NAT contributions. These deviations are by definition second order, but that does not make them unimportant. And the take home is to be thoughtful about the available degrees of freedom in your data set. And also, as Jeremy talked about this morning and came up in discussion, the basis vectors that you use in your inversion should be specific to your need. So if you're working, well, I won't go into the details, but you know what I'm saying. Like, you don't want to use a 20 phytoplankton groups and you know that 17 of them never exist in your place if you're trying to invert. You probably don't want that many anyway, but dramatic example. Okay, so let's extract pigments from hyperspectral information, measurements. This is kind of the start of this kind of work, the earliest efforts, well, successes. These are particulate absorption measurements from filter pad. So laboratory measurements, particulate absorption, Hofner and Sathyandranath, and they used Gaussians to represent pigment absorption. And to see how different pigments are contributing to the total phytoplankton absorption spectrum measured in the lab. We expanded that to ACS data, which isn't just phytoplankton absorption, but is particulate absorption with the NAP signal when we measure using our differencing method, like the inline here. So we do have to account for NAP. This is the open ocean sample, there's very low NAP, so this pulls out a low signal here, but again, we're representing pigments by all these different Gaussians, and the locations of them are determined by the known absorption of different pigments. And we do, kind of getting back to Raphael's question earlier, we do allow these to wiggle a little bit, but they're not allowed to, in the inversion, the Gaussian locations and the widths also, it's not free to fit as it, with no boundaries, because otherwise it would fit exactly. And these are set based on where we know the pigments absorb. So we're trying to get the information on these pigments out with a little bit of wiggle room, because maybe the pigment maximum absorption wavelength changes because of something in the cell, but they're fairly well constrained pigment absorption spectrum. And from that type of inversion to get pigments from ACS, then we can start to expand our in situ data set of pigments. This is chlorophyll A, but then we can also look at chlorophyll D and B and start to see patterns and accessory pigments with in situ data that's going to enable us to build algorithms with many, many data points for hyperspectral remote sensing. And importantly, we need to look at ratios of pigments, because this is an example of states E to C, but we also look at all the accessory pigments relative to chlorophyll A, because most pigments are correlated with chlorophyll A. This is the data set on the top from Sasha and colleagues, and you can see total chlorophyll A along the top of this correlation matrix, and all the accessory pigments, and nearly all are pretty significantly correlated. So if everything is correlating with chlorophyll A, it's important to keep in mind that we might want to look at ratios to see relative changes in pigments. Chlorophyll A, yeah.
Is that correlated with HPLC chlorophyll A or other methods of chlorophyll A about?
This is all HPLC in this slide, but if we make a scatter plot between this chlorophyll A and chlorophyll B from the ACS, they would also be correlated. Yep. Chlorophyll C and chlorophyll A tend to be more tightly correlated because C to first order is in the bigger stuff. But also, maybe I should have touched on this earlier. All phytoplankton have chlorophyll A, except for chlorococcus that has divinyl chlorophyll A. Let's just say all phytoplankton have chlorophyll A. All the other pigments are accessory pigments. Maybe does this come up in a lecture in week 1? Okay. Okay, good. So, the accessory pigments are passing the photons to chlorophyll A. It's actually doing the photosynthesis. So we really want to know the presence of the accessory pigments relative to chlorophyll A.
What's going on? Sorry, what's going on the bottom left-hand corner of the plot?
Yeah, so divinyl chlorophyll A and divinyl chlorophyll B and zeaxanthin are pigments found in the cyanobacteria that are inversely correlated to chlorophyll A from the ligotrophic ocean. So you have more of these prochlorococcus pigments when you have or cyanobacteria pigments when you have low chlorophyll.
I would like to know the difference between chlorophyll A and divinyl chlorophyll B.
There's a slight difference in the molecular structure that I don't know off the top of my head. And I don't know why some of them have evolved to have divinyl chlorophyll A, maybe. Yeah, I don't know, so I won't make something up. But the difference is, uh, a small change in the molecular structure, but serving the same function in the cell to do photosynthesis and, yeah.
But we have a plans for these could be hydros by environment.
Yes, so often when we say chlorophyll A, we actually mean monovinyl chlorophyll A. So those are. Just saying chlorophyll A is a little loosey-goosey. Well, often in the community, you just hear chlorophyll, and we really people mean chlorophyll A. But, yeah, if you have a data set that only has divinyl and monovinyl, you could add them. If you're just looking at total biomass. They're different, or you could just look at monovinyl as what we would maybe call chlorophyll A. Yeah, because that's what's most plankton have though. Yeah, you could also just check out your data and see, I would, depending on where it's from, but I would expect it to be the monovinyl chlorophyll A concentration to be much higher. Yeah, it's a good check. And okay, so now we've got our pigments. Well, I guess also, yeah, so pigments are found in different groups, but also just a reminder that they're really not diagnostic. Maybe there is some utility to separate groups based on pigments, but it does have limitations. Okay, so now we've estimated pigments from particulate absorption, but we want to move to remote sensing reflectance. These are the same equations you saw this morning from Jeremy, but now instead of a basis vector for phytoplankton absorption, when we do our inversion, we're going to replace our one basis vector with a dozen, say 10 Gaussian functions. So our phytoplankton absorption will be represented by that series of Gaussian functions like we saw in the ACS plot. And when we run this inversion, we do normalize by the uncertainty, and this actually results in significant, noticeably better results. So when you fit your cost function to find the best difference between your measured and your modeled spectra, normalizing by the uncertainty means that the inversion will have more leeway, let's say, where you have higher uncertainty. So it's optimizing the fit, but it can be the measured and modeled difference can be larger where you have higher uncertainty. That makes sense. And we use wavelengths between 400 and 600 where we have good signal.
Sorry.
It's all good. So using these spectra, which are collected at these locations around the world with a hyperpro and buoy mode. So we have our remote sensing reflectance at the surface and using coincident HPLC pigments measured at the same locations. First, we run the inversion that includes Gaussians to represent pigment absorption. And then we relate the magnitude of those Gaussians to the concentration of our HPLC pigments. And for the RRS spectra, we're using absorption by chlorophylls B and C and carotenoids in the blue, in the 400 up to 500 kind of wavelengths, where we have a lot of signal from remote sensing reflectance. And so we see correlations between Gaussian absorption at different wavelengths in the blue from our inversion and concentration of different pigments across a broad type set of data from different water types. That's one way that we try to estimate pigments from hyperspectral. Did Sasha talk about this already? Okay, so Kramer et al. And don't worry, we're not enemies. Everyone sometimes is concerned that we both have a pigment algorithm, but we're friends. Um, yes, so Kramer and colleagues developed another method to estimate pigments from hyperspectral using this data set of coincident RRS and pigments from around the world, again in situ, hyperpro, buoy mode, HPLC from these cruises. And this is a method that determines the residual RRS by differencing the measured spectrum and a spectrum defined using forward modeling. So like hydrolite, but actually much simpler, just using sort of broad brush, global average basis vectors for our IOPs. They forward model RRS and difference are measured and modeled. We don't expect them to match, and we're not trying to forward model RRS to match. We're trying to forward model a approximated RRS based on average inputs and then difference it. And the residuals are going to tell us, oh, there's something, because if our basis vector for phytoplankton absorption was a smooth one, like Jeremy showed in his plots, just this classic blue peak, red peak absorption. But if there's a difference in absorption in a given sample due to some anomalously high accessory pigment, it will create a very subtle feature in the absorption spectrum that's propagated to the reflectance. So when you difference, you see these residual features that are only possible to get at with hyperspectral and might tell us something about the pigment absorption that's changing very subtly our remote sensing reflectance spectra. And so then looking at different derivatives of these delta RS spectra, they related the value and also for different derivatives of the spectra to HPLC pigment concentrations. And in this method, they're extracting 12 or 13 different pigments. So trying to relate modeled and measured pigments using that residual approach. Now you have maybe 13 pigments. How does that tell you about PCC? Circling back to the challenges there, going from pigments to PCC, it's non-trivial. Sasha's done a lot of great work. It has multiple papers I encourage you to look at. But they use a clustering approach to determine how many phytoplankton groups they can separate using their pigment data, and then kind of these broad brush broad taxonomic categories that are associated to the clusters that are determined mathematically. So these clusters fall out just based on this analysis, and then we see that these pigments are found in haptophytes, these pigments are found in diatoms, not uniquely, but there are some unique groups that are being separated with the clustering. Okay. Now we've estimated pigments a couple of ways. Going forward, I'm going to show you some demos on satellite data using the method that I developed because that's the code I have. But in the future, the goal is Sasha and I are both working with NASA to have these pigment algorithms implemented, so you will be able to access both data from both methods at some point. So looking at paste data, here's chlorophyll from a swath on the West Coast. This is the Gaussian method to estimate pigments. Now we're looking at chlorophyll C and chlorophyll B. This is a case where we don't see correlation that's uniform across all pigments varying together, but we actually see some complementary concentrations where you have low chlorophyll C with high chlorophyll B. No idea if these are correct, but the algorithm to invert the RRS and pull out pigments is identifying something different about these different water masses within this total chlorophyll. So as part of a major effort by dozens of paste validation teams, there are people all over the world, including Trevor's advisor and actually lots of people in here and their advisors who are collecting data for NASA to try to start to evaluate and tune these algorithms. So you all can use the data. Another example from an eddy, a Gulf Stream eddy, a paste chlorophyll image, chlorophyll C from the pigment, Gaussian approach, inversion approach, and then the ratio of C to A, where maybe you see that around the eddy, there's higher relative chlorophyll C, could be photoacclimation, could be phytoplankton community. But we're starting to be able to tease out something different in the pigment, in the pigment absorption. I mentioned this earlier, but the emit sensor is used to study geology, but it does take some, it's hyperspectral and visible, and it takes some images over the coast, so I snagged some data to play with in the Puget Sound. There's only like 2 images, but still cool. The spectra looked good. So here's an example of the NASA default OCX algorithm calculates chlorophyll. This is the that I applied to the emit data. And then the Gaussian version of the algorithm, which maybe just as a first pass looking at it, it looks like this algorithm, the OCX, is picking up a lot of the river mouth here, a lot of CDOM maybe, but when we're looking at pigment absorption just in the blue, perhaps not assigning that CDOM to chlorophyll A, so.
So when you're doing these inversions, are you still keeping the same number of typical IOPs that we're using, or are you adding in the additional pigments for this inversion?
Yeah, so it's the same number of IOPs in that it's always CDOM absorption, detrital absorption, phytoplankton absorption, water absorption. Those are our 4 absorptions. And then backscattering by water and backscattering by particles. Those are 2 backscatterings. But the pigment absorption is not one basis vector, it's all those Gaussians. And that's possible because we have hyperspectral, this is done with hyperspectral data. So there's, in theory, enough degrees of freedom in those 150, whatever, 80 wavelengths to invert for the magnitudes of the 13 Gaussians, yeah.
So then, do you have to apply a ton of regularization, or is it Gauss? Are you not regularizing that inversion? Like, kind of constraining with an L2 or L1 norm at the end of the objective function.
I don't know the question.
I guess I'm just wondering, as we add in more of these priors,
Yeah.
are we pushing our inversions in one direction or another that needs to be corrected with some other damping force? That's
I don't know. Like as you have more and more basis vectors you're including, is it possible that it's gonna suddenly swing and attribute, like one of them is gonna get loaded much higher than is realistic and we need to constrain that? Is that kind of the question?
almost, yeah, because I'm looking at like these axes, and one's like 10 and one's 2, and obviously it's not apples to apples, but just wondering, like, in those selections, if that's how that's changing the parameter space. And how
Yes.
that's then changing.
Okay, I think I see what you're saying. So like when you add more, then you invert, then you've attributed like relatively less, let's say to the chlorophyll A absorption peak.
Right.
Yes, got it. Sorry, it took me a minute. Yeah, that's a good point. And there's a piece that maybe I got, well, with the scatter plot where now you have an empirical relationship between the magnitude of the Gaussian and for that number of Gaussians. And a HPLC in situ chlorophyll. So I'm not surprised by this difference because the inversion isn't tuned for this area. So maybe there's a different relationship between variability in chlorophyll and the Gaussian magnitudes in these super high chlorophyll waters. So the coefficient to go from Gaussian magnitude to chlorophyll is not also unique data. Wouldn't necessarily say this one is right either. But yeah, I see your question, and I think that what would be important is if you change the Gaussians, you then wouldn't want to use the same coefficients to go back to chlorophyll concentration, because that empirical relationship might change as they're loaded differently. Does that make sense?
Yes.
Okay.
Yeah.
Okay.
We're gonna play around with this a lot in the lab. They're gonna add more basic sectors and might see
Perfect.
instability. The more instability.
This is just me goofing around. Well, it's for proposal. out.
Then you really want. Yeah.
Yeah. Exactly. See how much better? And for the same emit data, I calculated B and C for fun. These are ratios to A. And I just think it's so cool because I don't know if it's accurate for C and B ratios, but there's obviously some water mass that has a different optical signature that's coming here. And you know, with in situ data, you could test in C, or with phytoplankton community data, you could evaluate that further, but it shows promise, right? It's not just noise static, TV static there, and TV static is a dated thing to say. You know, the antenna's a little crooked and staticky. Okay, take home message. I think you're all on it, but the question is not as simple as how much information, but which methods and approaches are relevant to your research questions. What are you trying to get at? And how can we incorporate other ancillary data, environmental parameters, polarization, being aware of overtuning and overtraining your model. At the same time, you want to take all the information you can for your question and throw it at the problem. Okay, I think that in the interest of time, I'll skip ahead. Unless we want to like take a little break and come back, but I mean, that's the bulk of it, I would say. There's a little bit of like new work tidbits at the end, but maybe what do you think, Patrick? For how much is left? 15 minutes. No,
I think we do it now and then we'll take a break and then we'll go that time, we'll be long.
okay. I don't want to cut into the lab time. Okay. Maybe 10 minutes.
For a break, we can take a break now, finish up, go straight on that.
We'll do 10 minutes, 10 more minutes. Sorry, I got excited and added a lot of students. Okay, recent work, which obviously you just saw some recent work, but here expanding even a little bit more, thinking about PCC. There's an explosion of new technologies. They're very exciting. There's tons to do. Lots of new ways to quantify plankton in situ. IFCB. Soon we'll see our data from Tuesday. But you know, these tools are powerful, and we're starting to accumulate in situ data that really expands our ability to work on algorithm development. Machine learning, obviously a huge yeah, Patrick's gonna give a lecture on machine learning next week more broadly, but plays a role certainly, especially in the kind of IFCB data that we collect, because we need to classify millions of images more efficiently than one at a time. So one way that we're trying to get at PCC from PACES and from remote sensing is with diatoms that we measure using IFCB data. And when we look at data from several different cruises here put together, we see that diatom carbon as measured by the IFCB is correlated with changing chlorophyll A. This is measured PCS stations. This is all throughout the North Atlantic, but different seasons. But what I want you to notice here is that there's a lot of variability around this relationship. And the other lines on the plot are previously published relationships when we're defining diatoms just using pigments. And some of these bias is not too unexpected based on how we define diatoms with pigments, but having this kind of imagery data in situ really starts to give us new information about relationships between PCC and, for example, chlorophyll A. going a little further there, now you can see this is the same information, but with actually more and more cruises added. And the distribution of diatom carbon calculated from different cruises and different ocean basins. So we're really starting to amass in situ PCC data that's highly diverse spatially and seasonally. Here's a map of those cruises where we have in situ IFCB data and also ACS everywhere at the same time. So maybe we could build some relationships between environmental and optically derived parameters in our diatom carbon. So this is ongoing work that our group is doing, that I'm doing. And in this case, we're trying to estimate diatom carbon in situ, but using our accessory pigments from optics derived in situ with ACS, but in case with RS, obviously, as an input. And so the target now is IFCB. And having IFCB data lets us use imagery as the target, which means that we don't have to define the groups using pigments. And now we can use the pigments as an input. So maybe the pigments in the water are correlated with the groups where we define the groups from the IFCB. The trained model, this is a random forest model. Happy to talk about this offline too. We'll just go fast for time. And then we've tested deploying it with some PACE data. So these are two PACE derived diatom images, one from the Gulf of Maine and one off the coast of France. And we don't have the chlorophyll plot here for comparison, but the patterns in diatom carbon concentration are to first order tracking the chlorophyll, but then you start to see different patterns in diatoms that only emerge, and we have this kind of algorithm that's not just relying on a direct correlation between diatoms and chlorophyll. And on this side, there's some in situ IFCB data thanks to Guillaume. And the PACE data is from one day and the in situ are from several, so we don't expect them to match exactly, but it's just a very first effort at comparing PACE diatoms to in situ diatoms, and I'm excited to keep going down that path as we have more coincident diatom data. Maybe we had such clear skies, we could do diatoms from Tuesday from the IFCB, calculate PACE diatoms, and see how our maybe we'll have a perfect R squared with our 3 data points. Actually, if we have 2 data points, we'll definitely have a perfect R squared. So hopefully only 2 stations have good data. Okay, and just finishing up with the messaging of how important uncertainties are, which I know you've heard, but also how important our data are in their representation of the globe, let's say. Gray shows the distribution of chlorophyll from satellite. Blue shows our in situ distribution. So even though we have got thousands and thousands of data points, they're not mimicking, they're biased towards interesting places with lots of plankton and chlorophyll typically, for good reasons. We're studying those places every once in a time. He wants me to go faster. But when we look at the temperature salinity plot, you could see, maybe we're missing some of the water types on the globe, and that's why we can't stop making in situ measurements. Keep going to see. And we have to propagate our error through these different approaches. So considering all the errors that go into our calculation of different parameters, and in this case, this is for the 2022 paper, we're estimating diatoms. We come up with 65% uncertainty, or 1 milligram per cubic meter. The maximum of those 2 is the uncertainty. What is it good enough? Who could answer that? I don't know. You, me? I mean, the answer is really maybe. Depends on what you need it for, right? That's related to the uncertainties, but for time we'll go on, but that's another nice paper from Sasha, and I'll have all the references for you all after this. And then, yeah, how can we, kind of ending thoughts, how can we navigate this untapped potential, but also with all these challenges that I've kind of focused on today, and yeah, important to consider different needs for different projects and what do we want to know and why, and consider scales of variability, and remember that uncertainties complete the data. And if you see a plot without error bars and you are looking at a study and there's no mentioned uncertainty, or you generate your own, maybe, try to revisit that and think, well, what would it mean if the uncertainty on this was really large? How would that change my interpretation of the results, especially comparing things? I mean, fundamental, good science. A nice commentary paper in EOS, I think, talking about overcoming the challenges in ocean data uncertainty, and just emphasizing how critical it is. So I think you've got the message. All right, I'm gonna leave the last few slides for you to look at in your own time. I think we've covered them all. Major challenges, and got to end on an up note, exciting opportunities. There's obviously lots to do, so if any of this excites you, that's great. We need more folks working on it. And in the spirit of Kurt Mobley, I'll leave a photo with you all from my optics class in 2011. Here's the one on Dr. Wardell and his children who are now in college. Sorry, I'm not trying to call you out of age. Mr. Wardell. But yeah, some of these folks are, I don't know, maybe even your advisors, but colleagues and.
Right.
Yeah, they're my TA. My trusty TA, Meg. But yeah, it's really an exciting opportunity that you all have to build your cohort, and I collaborate actively with multiple people in my cohort, and that's really a pleasure. So obviously stay in touch with each other and go out back to your labs and out in the world and share the love with other people too. There's a lot of folks who want to be here who can't because we don't have the space. So, welcome all into the fold as you take your optics knowledge out into the world. Thank you.